6 10 तक ज्वाइन करेंगे महेश राव सर, बात हुई है अभी। रोहित जी सुन पा रहे हैं आप? नंदिनी,
हाँ जी सर।
6 10 तक ज्वाइन करेंगे।
ठीक है सर।
गुड इवनिंग सर।
Good evening. I am audible.
Yes sir, you're audible. Thank you sir so much for joining.
Thank you so much. Sorry, it was so hectic schedule nowadays, so it's very difficult for me to get time. But anyway,
जी जी जी,
uh,
थैंक यू सो मच सर।
okay.
सर स्टार्ट करें?
Yeah. So,
नंदिनी, आई थिंक वी शुड स्टार्ट नाउ।
Okay, ma'am, am I audible and visible?
यस, गुड
should I
टू
share
गो।
the slide?
यस सर।
Okay. I think is it visible now?
यस सर। यस सर।
पीपीटी मोड पर कर लीजिए सर।
हाँ, मैं कर रहा हूँ, कोशिश कर रहा हूँ थोड़ा. Now it is okay.
जी सर।
Yes. So,
Oh,
greetings everyone. A very warm welcome to all the participants to the day 17 of Krishi Navyug 2026. I am Nandini Sharma, co-founder Agriculture Valley Research Foundation. Feeling truly honored to welcome you all once again to this platform where learning meets real world agriculture innovations. Krishi Navyug is not just a lecture series, it is a movement to connect students, researchers, and experts across India on one common platform of growth and knowledge sharing. Today, we are extremely privileged to have with us a distinguished scientist whose work is contributing significantly to the future of Indian agriculture. Please join me in welcoming Dr. Mahesh Rao, sir, Senior Scientist at ICR National Institute of Plant Biotechnology, New Delhi. Sir has been associated with ICR since 2011 and has built an exceptional career in genetics and plant breeding, focusing on improving crop resilience and productivity. His research primarily focuses on climate resilient crops, heat and drought tolerance, disease resistance, and yield improvement in rapeseed mustard. Sir has also gained international research exposure as a Serb Sire postdoctoral researcher at the University of Bonn, Germany, working on advanced breeding technique. He has led and contributed to multiple prestigious projects funded by DBT and DST and international collaborations showcasing his excellence in scientific research and innovation. With numerous research publications in reputed international journals and recognition through several Young Scientist Awards and fellowship, Sir continues to inspire the next generation of agriculture scientists. So, your work in strengthening crop resilience and advancing plant biotechnology is not just a research, it is a contribution towards securing the future of agriculture. For all of us, it is truly an honor to learn from your experience and insights. With great respect, I now invite Dr. Mahesh Rao, sir, to kindly take over the session and share his valuable knowledge with us. Over to you. The session is yours, sir.
Thank you, Nandini, for the wonderful introduction. Though it's exaggerated version of me, but thank you. And hope it's visible. The slides are visible. Is it?
Sir, it's showing resume the slideshow. Rohit, please check
Okay.
karo, sir.
Hello. Hello.
Yeah,
Sir, can you please stop share once and then reshare again once? Yeah.
that I can do. Wait. I will do it. Hmm. Wait a second. I'm doing that. I think multiple windows are open, so it's maybe hindering in the
Uh,
presentation.
maybe.
Wait, I'm doing that.
So just do one thing, close the PPT once, then reopen the PPT, stop share also.
Yeah,
Right?
I did. So
Yeah,
now
it's
it's
loading.
visible.
Yeah, it's loading. Yeah, it's so please like in PPT mode, do it in PPT mode.
So is it okay now?
It's not in PPT mode, sir. Can you move one or two slides at once?
Yeah, you can check it. It's moving.
No, sir.
Okay, wait. So I have to share a whole
Whole screen. Yeah, okay, you
screen,
can
I
do.
think. Yeah.
Mm, mm.
So wait. Hmm. There are multiple options. Screen one, screen two. I don't know.
Whole screen, sir.
A whole screen. I am finding that one. It is not available. There is two screen it's given here. So should I share screen one? When I can check it. Now it's visible
Loading,
and
sir.
move. Okay. Yeah, here it's moving fine.
Yeah, sir. Your screen is there.
Yeah, it's moving.
Yes, sir. Like
Okay, so
Sir,
Thank you.
it's
Yeah,
uh
it is in PPT mode, presentation mode,
one second, sir.
because here it's working fine.
नॉट इन पीपीटी मोड सर. इट्स ओके सर, आप अगर ऐसा कंफर्टेबल है, अगर कुछ ग्लिच आ रहा तो आप यही ऐसे कंटिन्यू कर दीजिए, लाइक विदाउट पीपीटी मोड.
Yeah, it's moving now.
यस सर, मूविंग.
Okay, so good evening, everyone, and thank you for hearing me, having me here. And thank you, Pragati, for the invitation. So my work is mostly on Brassica, and that is also on pre-breeding. So I don't know how many of you are from agriculture background or genetics and plant breeding. So I am working here from 2011. I joined the institute. So my topic will be the pre-breeding efforts using the CWRs, that is the crop oil relatives, and parental diploid progenitor species to broaden the genetic base for future brassica crop varieties. So if you know about the polyploid crop and mostly for the MP diploid or allotetraploid crops, there is problem of genetic diversity. So genetic diversity is very low. So how we can increase that one, that can be helpful for the breeders to develop the elite variety, or they can get maximum gain, genetic gain, or they can get the more heterosis for the development of varieties. Also, if we have the crop oil relatives, they are having a lot of traits that we can transfer from wild to the cultivated one. Same way, the parental diploid species, they are having many traits which is very useful for the breeding purpose, that we can transfer through the pre-breeding efforts. So my whole presentation will be on the pre-breeding, so bear with me. And if you are from the agriculture and genetics and plant breeding background, so you will love to learn the things whatever I'm going to tell. And if you are from the other subject, you can enjoy the seeing the pictures. Different color pictures are there in my presentations. So the outline of my presentation will be the Brassica species. Firstly, we will know, and what are the interrelationship between different species we are having in our U triangle, and also how pre-breeding we are doing at NIPB. So we will learn this, and how it is helping for the to the breeders, and what we can do through the pre-breeding. Also, we are having some wild species Brassica that we are characterizing for different traits that I will show you here. And then three different techniques we are using in the pre-breedings, tools you can say, yeah, methods you can say. So firstly, we are utilizing the parental species to resynthesizing the Brassica gentiana, that is the yellow tetraploid. Also, we are introducing a trait from the wild species to the cultivated one. And also one novel approach we have started 4 years back, and with the help of BARC Mumbai, where we are mutating the F1 MP haploid to generate more diverse MP diploid Brassica gentiana, that I will show it here. Then we will I will show the some promising lines we have identified for the different biotic and abiotic stresses from our research as well as from our collaborators. Then we will draw a conclusion on the basis of our whole presentation. So if you'll see the oilseed Brassica, so recently it was ranked first in the oilseed production in all the vegetable oils which we are growing here. But still we are importing huge quantity of vegetable oil from different countries to fulfill our domestic demand. So there is need of or that we can uh, develop the variety, or we can harvest maximum potential from our germplasm to develop a good variety that can give more productivity, and that can sustain in the different climatic conditions, and that can go lead to the production of more oilseed crops in India, that we can minimize our import or dependency on the other country. So also with the same way, there is a question that are we leading towards our goal of sustainability? Because we are using a lot of chemical fertilizer, a lot of pesticides to control different biotic and abiotic stresses, biotic stresses especially. So there is need of our that we can explore the different species, different germplasm to identify the genic kind of resistance for different biotic and abiotic stresses, that we can produce more in the changing climatic conditions also. So, if we talk about the domestication, you all know that due to the domestication and due to the development of elite varieties, the alleles were seeded off during the process of evolution, during the process of development of different varieties, elite varieties. So that alleles we are having in our either land races, in our progenitor species, as well as in our wild species. So that is the need that we can bring that back, and that can be characterized and that can utilize in our breeding program to strengthen our mapping that breeding population, and that can be more important to accumulate more positive alleles to generate the develop the elite varieties to get the more production and even further different to further resistance to the different biotic and abiotic stresses. So we can explore the wild species, we can explore the land races which is available, we can explore the different exotic and indigenous germplasm, even we can explore the parental species from where the modern crops were derived, and that can be characterized and that can be bring back in our cultivated gene pool that we can increase it and we can utilize for our breeding program. So if you'll see that U triangle, if you are from agriculture, as I told you all, so if you'll see, this is the U triangle. It was given by Nagaharu Yui in 1935, where they have demonstrated the different relationship between the different diploid and tetraploid species. So if you'll see, this is the Brassica gentiana, and if you'll see the Brassica gentiana, yeah. So oh, it is visible now because I have transferred to the presentation mode now. So it was derived from the Brassica nigra and Brassica rapa. These are the diploid species. So if you'll talk about this Brassica, because in India we are growing Brassica gentiana as a oilseed crop, but in Europe, in other countries, the Brassica napus is the major oilseed crop in compared to the Brassica gentiana. So we are mostly focusing on the Brassica gentiana, how we can maximize the genetic diversity. So if you'll see that these three amphi-diploid species or allotetraploid species, that is Brassica gentiana, Brassica napus, and Brassica carinata, these were developed in nature, yeah, evolved in nature from the interspecific hybridization and chromosome doubling. There are two or three theories are given where that how these amphi-diploid species were evolved in nature from the diploid species. So for the Brassica gentiana, Brassica rapa, and Brassica nigra was the parent. For the Brassica carinata, Brassica oldacea, and Brassica nigra were parent. And for Brassica napus, Brassica rapa, and Brassica oldacea was the parent. So whenever you are thinking that it's very difficult to evolve the for the evolution of any yellow tetraploid species or yellow polyploid species in nature, and there is very less chances that very few germplasm from the both parent or three parent or how many parents it's involved from different genomes, different species that was involved for the evolution. So the rest of the population from that diploid species are not included in the developmental process or evolutionary process of that polyploid species. So that is also having very good alleles or genes or traits that can be utilized through the pre-breeding, and we can harvest it and we can put it in our cultivated germplasm. If you will see the matting system, the most of the diploid species are self-incompatible except the Brassica rapa yellow sarso, and the tetraploid species are self-compatible. And also, if you'll see the pre-breeding, what pre-breeding refers to, all the activities to identify desirable characteristics or genes from the unadapted materials that cannot be used directly in the breeding program, uh, as well as to transfer these traits to an intermediate set of material that breeder can use the further in the varietal or crop improvement program. So the what breeder exactly do, they use to generate the breeding population or intermediate set of breeding population that can be characterized and transferred to the breeders that they can utilize in the breeding program for the development of elite varieties. So why it is important in Brassica? and Brassica gentiae especially. So there are very less genetic diversity in the cultivated MP diploid species, so that's why it's needed. There is genetic ceiling for different economic traits like yield contributing traits, biotic and abiotic stress tolerance, or also for the if you are looking for the CMS restorer system, you have to look for the wild species. Also, the diploid and wild and related species are the reservoir of the wide range of economic trait that can be transferred to the breeding program. So if you'll see here, the what is the concept for the doing the pre-breeding. So if you'll see that this funnel, this is the all the genetic diversity which is available in our primary gene pool that breeder can utilize for the breeding program. But in case of polyploid or yellow polyploid species, crops, this funnel is already not fully filled. So what we are doing, we are using different methods that we can generate the genetic stock that can be characterized, and we can fulfill that funnel that breeders can utilize for the development of varieties. It was very interestingly described by Sivali Sharma et al. In 2013. They have published one review article where they have described very nicely what is the pre-breeding and what is the crop improvement and how they both are linked together. You can go and search for that paper and you can read if you are interested to learn more about the pre-breeding. So in Brassica mustard, what is the scope and benefits for the pre-breeding? If you'll see, we can resynthesize the amphidiplloid species. We can go for a targeted new plant type development. We can go for development of new synthetic species. We can go for transfer of genes from CWRs to the cultivated species. We can learn some scientific advancement. We can learn some of the basic tools, basic basic biology behind the development of polyploid species from the diploid species. We can broaden and improve the gene pool of primary species. Also, we can go for the QTL mapping for the economic traits, identification of novel traits. We can go for the varietal improvement, and then also we can go for the study through the evolutionary process. Even in the nature, hexaploid brassica is not available, but some of the scientists, they have accumulated all the three genome, that is A, B, and C, and they have generated the hexaploid, which is not available in nature, but they are trying to test that the suitability in the current scenario. How if you are going to combine all the three genome, and if you will develop the hexaploid brassica, is it going to helpful for the farmers and the breeders or not? So they are testing for that one also. So pre-breeding, what we are doing in rapeseed mustard at ICR and IPV. So we have characterized the crop oil relatives in brassica. Then we have resynthesized Brassica gentiae from progenitor species. Then we have developed the mutant resynthesized Brassica gentiae lines. Then during the course of our wide hybridization process, we have identified and registered one genotype which is very good and which is having the wide compatibility traits, that is named as the NRCP Virapa 8. And then synthetic amphidiplloid we have developed through the using the bridge species of Brassica rapa, and to transfer the genes from the CWRs to the cultivated Brassica gentiae. So we have developed the synthetic amphidiplloid, that is the transient kind of species, synthetic species that can be utilized for the transferring the trait. And then advanced introgression lines we have developed for the alternaria resistance. That resistance is not available in the primary gene pool. So that we have transferred, and we have identified some of the introgression lines which is performing very well. Then also whenever we are going to talk about the pre-breeding, there are a lot of linkage drag, because whenever you are going to cross with the unadapted materials or wild species, a lot of unwanted genes or segment of the chromosomes comes in our population. So how we can remove that linkage drag if it is associated with some closely associated with some of the economically important genes. So the one methodology we have targeted, and that is we can target in our try in our pre-breeding program to remove the linkage drag. So firstly, coming to the characterization of the crop oil relatives of brassica for morphomolecular diversity. So you can see how many species and how much diversity we have. So these are the species which we have at our center, and we are characterizing it for the different biotic and abiotic stresses, and we are trying to utilize these in our breeding program to generate the pre-breeding materials that we can share with the breeder. So you can see the potential in the wild species that we can target for our development of genetic stocks. And if you'll see the distribution of these wild species which we have at our center in the different part of world from where it belongs. And also you can see that we have done the genetic and morphological and molecular diversity, as well as we have checked for the different botanical traits for these all the wild species, and also we have checked the seed size, oil content, even we have went for the fatty acid profiling to see if there is some valuable traits is available that we can harvest apart from the increasing the genetic diversity in the primary gene pool. And this paper was in is under review and hope it will come soon. So once we have characterized for the morphological and molecular diversity, we have thought for the characterizing for the biotic and abiotic stresses. We have made one panel of genotype, including the wild species, cultivated species, land races, and even the some interrogation lines we have included. So we have targeted three strategies, like for abiotic stresses at germination stage, juvenile stage, as well as the reproductive stage. So it's not necessary that the genotypes which is giving the resistance for the germination stage is going to give the resistance for the terminal stage or reproductive stage. So we have characterized these all the lines or species for the stress tolerance for the heat and drought stress at different developmental stages. So for germination stage, we did in vitro condition. For heat stress, we use the BOD, and for the drought stress, we use the PEG, PEG mediated drought conditions, and we have analyzed some of the morphological data as well as some of the biochemical data, and we have come up with the some of the genotypes for the heat and drought stress, which is giving very good result as compared to with the U triangle species, all the 6 species. So these species is having the potential that can increase the tolerance for the germination level heat and drought stress in the cultivated species. So that can be utilized for the breeding program if necessary, and we can go for the pre-breeding for transferring these traits from these wild species. Same way for the seedling stage, means juvenile stage, we have identified, we have taken some of the data for the morphological as well as biochemical data, and we have analyzed the data, and we have found out that these are the species, these are the lines which are giving very good result in compared to the all the 6 U triangle species. So if you want to target some traits that we want to transfer from wild to cultivated, we can go for this one. If we can make the process and we can transfer to our uh, primary gene pool for the enhancing the heat and drought stress at germination in juvenile stage. Same way for the reproductive stage, we have done the drought stress screening under runout shelter condition, and you can see we have identified some of the genotypes which are very good in compared to the cultivated species. Same way we have done the molecular analysis of these wild species, including the land races, and you can see the diversity which we are having at our centers. And also for the heat stress we have done, and still the data is we are analyzing. Same way, one of our collaborator from IRI vegetable science division, he has screened for the Alternaria brassicola, because in Brassica oleracea, Alternaria brassicola is the major disease, fungal disease, and the resistance is not available in the cultivated species, that is the Brassica oleracea. So they have identified, they have screened all the wild species whatever we are having, and they have come up with some of the genotypes which is giving the resistance, and they are making the crosses, and they are trying to transfer that trait from wild to the cultivated Brassica oleracea, that is the cauliflower. Same way, one of the scientists from uh, entomology division in IRI, he has screened for the aphid, mustard aphid, for these all the wild species, and he has come up with the some of the promising aphid resistance species on the basis of the disease incidence and different parameters he has analyzed. So I am telling that these are the potential things. If you are want to screen, you can go for screening for all the traits, whatever biotic and abiotic traits are available, that we can find out some of the promising line, and that can be target for the transferring from the wild to the cultivated species. Same way, orobanki is a very good big problem, but nowadays some of the scientists have developed the herbicide tolerant brassica. But earlier we have done this work to identify the herbicide tolerance in our wild species collections, and we have identified some of the genotypes which are giving good tolerant in compared to the cultivated Brassica gensea. So that can be also targeted for transferring that tolerance from wild to the cultivated. But nowadays the scientists have developed some of the lines which is giving more good herbicide tolerance in brassica gentiana itself, so there is no need to transfer. If we already have the trait in our primary gene pool, there is no need to go for the wild species for the transferring that trait, because it's very difficult task to transfer the trait from the wild to the cultivated one. But if it is not available in the primary gene pool, then we can think on for transferring from secondary and tertiary gene pool if it is possible. Same way, we are having one panel of genotype from 161 genotype of brassica gentiana. So we were targeting for the seed number per siliqua to identify some of the SNPs and some of the genotype, promising genotype, which is having the more seed number of siliqua, and that can be utilized in the breeding program. So we have screened that lines, and we have done some of the SNP genotyping, and then we have done the GWAS analysis to identify some of the SNPs for the siliqua main raceme, siliqua length, seed number per siliqua. But still, we are trying to make the crosses and validate these SNPs in the population. That will be helpful for the breeders for transferring these trait from the these promising lines which we are having at our center. And also we have one panel of genotype for 285 genotype, which we have targeted for the heat and drought stress. And that was in one of our Biodiversity International and CEAT Alliance project, where we have made one panel of genotype where we are targeting to search the drought and heat tolerant lines which is performing well. So we have firstly started for the heat stress we have started from 285 genotype, then we next year we come up to the 37 promising lines, and our target was to come pinpoint at the 1 or 2 genotypes. So we come with the 5 tolerant genotypes which were performing very well. And the same way for the drought stress, we have started with the 285, then come up with the 57, and then finally we landed on the 5 tolerant genotypes. And these genotypes we have tested the 57 and 37. We have tested for all the 3 stages: germination, seedling, and reproductive stage, and we have identified 5 most good and tolerant genotype for all the 3 stages, and that can be utilized by the breeders for the breeding program, as well as we are trying to find out the molecular mechanism behind that, and we are doing some of the omics analysis, and it is underway. So we can find out some mechanism that what are the molecular basis of for tolerance for the heat and drought stages at different developmental stages. So this project is underway. So we are trying to do it, and hope in coming 2 or 3 years we will complete this project. So once we have identified some of the wild species for different traits, so how we can transfer to the cultivated species? That is the major question. So these are the steps which we used in the wild hybridization in brassica. Firstly, we attempt the crossing, then we go for the embryo rescue for the F1 development, because mostly we are doing the wide hybridization where the 2 genomes are involved, different chromosomes number are there for the different species. So the seed setting is very poor, or it's not possible to get the seeds, F1 seeds, in vivo seeds. So we can go for the embryo rescue, and luckily we got one wide compatible genotype that I will show you later, where we can make the crosses and we can get the seeds, so there is no need of embryo rescue. So that is very biggest achievement for us as a pre-breeder, that we can remove the embryo rescue protocols from our research program. Once we develop the F1, we can go for the pollen viability analysis. In most of the wide hybrids, the pollens are not viable. So we can go for the cytogenetic analysis for the classical as well as molecular cytogenetic analysis. We can do the ploidy analysis through flow cytometer. Also, we can confirm the hybridity. And then if needed, we can go for the colchicine treatment, that we can go for the chromosome doubling, that can the plant which we derived from the interspecific hybridization can be stable and can be fertile, that we can utilize in our breeding program. Once we do that colchicine treatment, we can do the bud pollination to generate the next self generations, and then for the selfing and selections we can do. Then we can do the characterization and stability analysis through the morphological, molecular, and cytological tools. And if it is the different species wild species we are using, we can, after the bud pollination, we can get the synthetic amphitriploid, and that can be crossed, and that can be back crossed with the different cultivated species, and then we can generate the back cross population, interrogation population, and that can be further characterized for as per our research plan. So in that method, we started with the resynthesis of Brassica gentiana in long back, and where we were utilizing the Brassica rapa and Brassica nigra as a parental species, different accession of Brassica rapa and Brassica nigra we used. We crossed it and we developed the F1, and then that F1 was treated with the colchicine, and then we did the bud pollination to generate the S1, and then subsequently we did the selfing and selection to generate the advanced generation. Also, we targeted like this three-way cross or two-way modified two-way crosses we have attempted, where we have crossed the Brassica rapa with the other Brassica rapa accession, like Brassica rapa yellow sarso we have crossed with the Thoria, and then the F1 we have generated, and that F1 was crossed with the for the Brassica nigra to generate the F1. And that MP haploid F1, if you'll see here in the down picture, where we have generated the F1 of MP haploid, and then we have done the hybridity confirmation of whatever analysis I have proposed to do in the F1s, and then we have done the colchicine treatment and bulk pollination to generate the S1 subsequent generations. And then in the last we have characterized, and these are the some of the lines, RBJ lines, which we have identified, which are performing very well for the different traits. And the red one we have shared with some of the genotype, some of the institutions for the breeding program, as well as some molecular biology studies for the different traits. Two strategies we have identified applied in this one. Like firstly, we were targeting to generate the advanced generations that we can stabilize the population. Then we can share with the different breeders or different stakeholders that they can utilize in the breeding program. But that take a lot of time, like 6, 7, 8 years you have to spend if you are growing once in a year crop. So it will going to spend 7, 8 years to establish that stabilize that genome or that population or that genetic stocks to characterize it. So the advanced generation we have developed firstly, and then we have analyzed this one from using the molecular and morphological tools, and we have identified some of the genotypes which is performing very, very well. Contrasting genotype we have identified in compared to the Varuna, that is the cultivated one. And if you'll see the pollen viability, that is also going to increase year by year. So in the in the S8, S9 generation, we got the more than 90% pollen fertility in the most of the genetic stocks. But it took a lot of time, 9 to 10 years you have to spend to get the S9, S8 generation to go further utilization in the breeding program. So also we have done some of the genotypic analysis. We have done the GBS analysis to identify to do the diversity analysis within the group, between the groups, and also we are targeting to see the intergenomic recombination. That work is going on. But what we thought that rather than going for the 7, 8 generation, 9 generation, we can characterize these lines in the F1, S2, S3, S4 generation, that is in the early generation of the resynthesized lines development, and that we can pick some of the lines which is performing very well for the different traits. We can share with the breeders, and that they can utilize that line in the early generation, and then they can do the selection in their own population. So that can reduce 4 to 5 years for developing the varieties. And what we got that in the initial years, initial generations, the diversity is much, much more than what we got in the advanced generation, morphological as well as the molecular diversity. Because whenever we are going to do the selfing and selection in the further generation, we are going to lose some of the alleles from our population. So that we can capture in the early generation, and that we have published in last year in the frontiers. And that is the one way where we can save some of the times by utilizing the early generation resynthesized line in the breeding program. Also, we have started with the induced mutation in the synthetic Brassica gentiana development. That was a new methods where we thought to maximize the intergenomic recombinations through the gamma irradiation. So firstly, what we did, we have standardize the gamma treatment for the doses, how much we can do for the MP haploid, because MP haploid is having one set of genome from A and B that is very sensitive to the any mutagen. So that will be good if you are going to treat with that one, and we can get some more recombination, and that is going to give the more diversity in our early synthetic Brassica gentiana. So firstly, we have standardized the dosage, and we have with the help of BARC, we have one partner from there, and the paper is under review. And once we have standardized the dose, we have treated that F1 with that mutagen, that gamma. And then we have developed the S1, M1, M2 generations, and then further we have developed the next generations. In the early generations, we have screened, we have evaluated for the morphological as well as molecular diversity. Also, we have screened these lines which we have developed from the synthetic mutagenesis. That is, we have tested for the heat and drought stress, because that was one objective of the project, and we have identified some of the genotypes, the genetic stocks which were performing very well in compared to our cultivated, which we are having as a check. So that we some of the lines we have identified for the heat and drought stress at this 20 stages, but still some of the genotypes are not stable yet, and we're trying to get the stability first, and then we are going to evaluate again for the different biotic and abiotic stresses to identify some of the genotypes. Also, these materials are very interesting, because if you'll see the diversity spanning the from all the population that we have developed from this mutagen experiment, that it is having a highly variation in compared to the our synthetic and other genetic stocks we have developed here. So this is a potential method where we can target to develop more diverse line in a different way by using the different mutagen treatment. So once we have identified the wild species, how we can go for the development of introgression line using the crop relatives. So there is two methods we can think on. That wild species directly we can cross with the MP diploid Brassica gentiana, but due to the ploidy difference, due to the chromosome number differences, it's very difficult to get the seeds or even the F1 hybrids even after the embryo rescue. So what we applied here, we have taken one bridge species as Brassica rapa. We have taken as a bridge species. We have crossed that one with the wild species. Then we have developed the F1. We have done the colchicine treatment. Then we have developed the synthetic amphitripla. That is the transient one. And then that transient one will crossed with the Brassica gentiana. So ploidy level was almost same. And the Brassica rapa was used as a bridge species that was crossable with the, because one genome in Brassica rapa is available already in the Brassica gentiana. And also it was crossable with the wild species. So we have developed we have tried this method to develop the introgression lines, and we have developed many synthetic amphitriploid species. These are the synthetic amphitriploid species with different wild species. You can see that these are the synthetic amphitriploid species in the right hand side of the arrow with the different wild species. And one of the successful example I will show here. And these are in the different generation of early alien introgression line development stages, and we are characterizing it in the each generations. And also whenever we are going to use the wild species, we need to confirm the introgression from the wild species to the cultivated one, whatever we are developing the introgression line. So for that, what we targeted, that we have developed the we have generated the genome sequence of the wild species, and we have developed the species specific marker. So as of now, we have developed for the 3 different species recently, and we have checked the specificity with the wild species, and that can be used for our introgression breeding to check the introgression from wild to the cultivated one when we are going to, because sometime if we are going to do the more back crossing, we are going to lose the wild genome. So that is also a risk. So we have to at least check that the whatever material we are developing, at least we are getting some of the wild introgression or not. And through these methods, we have done one of the we have one success story for that one, because we have developed the synthetic amphitriploid. Earlier when I joined here, I got that material, and that was crossed with the Brassica gentiana, and then we have developed the advanced introgression line. So you can see this is the rapa, and then the white one is the Diplotaxis erythroides. We have crossed it, and after embryo rescue and colchicine treatment, the Eru rapa was generated. That is the synthetic amphitriploid, and that's what further crossed with the Brassica gentiana. That is the RLM 198. And then back crossed with this RLM 198 to generate the introgression line. So you can see the ERJ 155. So now we are having 195 introgression lines, and that was we are screening from last 10 years to the different in the hotspot location as well as at ICR and IPB under control condition for the Alternaria blight. Apart from the genetic diversity, we have screened for the rigorous screening we have done for the Alternaria blight, and we have got the resistance lines. So if you'll see, these are the 15 lines which we got, which is giving the highly resistance reaction for the Alternaria brassicae. Also, we have analyzed for the diversity, for the morphological and molecular diversity, and we have identified some of the trait-specific lines for the ill traits that we are utilizing for the other program. But here the main focus was on the Alternaria brassicae to transfer the from the wild to the cultivated species, because in the cultivated species we don't have any resistant line. So we have done some of the histopathological analysis and biochemical analysis to confirm the resistance of these lines. And we have come up with some of the lines which are very, very good, which the further molecular biology work is being done by one of the scientists here at NIPB under one project from these are identified lines. So hope we will come up with some of the molecular mechanism, but still these are the lines we have shared with some of the centers to utilize in the breeding program to transfer the trait in the varieties, that the varieties can be resistant to the resultant area brassicae. Same way, whenever we are going to cross the different species, there is problem of compatibility. Luckily, we got one lines which were giving very good compatible reaction with the wild species. Earlier it was started with the Brassica rapa and Brassica nigra, and we are going for the synthesis of the Brassica gentiana lines. So one of my student is working on to underline the mechanism, molecular mechanism, what is the factors responsible for this one. So firstly, we did the analysis. So you can see how we did. Earlier, we have to do the conventional method using the tissue culture, where we have to do the ovary culture, embryo culture, then we can go for the development of roots, shoots, and then hardening, and then we can transfer to the pods that F1 can be developed finally. But now we can make the crosses by using this rapa with the nigra, and then we can develop the seeds and we can grow it easily, and then we can go for the colchicine treatment as per our experiment. Same way we are trying it for the wild species. And if you'll see the, and what is the beauty of this one, in almost all the research article, if you'll see for the development of synthetic ampidiploid or the development of synthetic ampidiploid like Brassica gentiana, Brassica neposa carinata, they have used the tissue culture for the developing the F1. But here we have found this one, whenever we are going to make the crosses with any accession of Brassica nigra, we got the seed. So you can see here in this graph, in the three graph given here, the first one is the NRCP rapide. You can see the success rate in the red bars, that in each and every combinations we are getting the seeds. But in other two lines which we used as a control, where it depend on the genotypes which we are using in the crossing program. But here you can see the frequency are more or less in the different crosses, but still we are getting the seeds in each and every cross. So it's very, very good. So you can also see the success rate in vitro and in vivo condition. With the normal, it's 2.71. And the total cross combination with the NRCP rapide 35.52. In vivo condition, it's 0.38 for the normal one, and for the NRCP rapide 30.46. So it's huge seeds we are MP haploid seeds we are going to get, and that is going to lead for the development of synthetic ampidiploid, that is the Brassica gentiana, which we are targeting here. So one of my students is working on that. She has done the pollen pistil interaction to see the when the embryo is going to abort in the non-compatible Brassica rapa, and that stage we can target for the molecular biology study, and she is doing her work in this area. So earlier we have to do the embryo rescue. There is now it's not required for our pre-breeding program here. So we can directly cross with the Brassica rapa and RCP rapide with the nigra. Then we can develop the F1, and then we can go for the synthetic ampidiploid development through colchicine treatment. So embryo rescue is not required. Same way we are targeting this NRCP rapide with the different wild species to check how much we are getting. And till date we have got from 7 to 8 wild species the seeds we got, and without embryo rescue, and that we are targeting to develop the synthetic ampidiploid. So that will be a very, very valuable material for developing the going for the wide hybridization program. Also, whenever we are going to do the development of polyploid species, there is a chance that you can learn or you can understand how the polyploid were generated in the nature. We cannot mimic that the whole process, because it's long process for developing from the diploid to the polyploid species and stabilizing in the nature. But at least we can understand how was the situation, how the chromosome was going to rearrange when those two different genomes are coming together. One is having its own cytoplasm, other is coming from the different cytoplasm, they are coming together and they are living together, and they are how they are adjusting. So one of my student is working on that. We have developed the different generations of the synthetic MP diploid. She has done the analysis for the morphological traits, as well as now she is targeting for the molecular traits. So this is the one we have analyzed. This was the funded by one of the project from the ANRF, and it's going on. So you can see for the one combination. So we have generated a lot of combinations for that same, but one combination we have got this kind of results, and we are going to analyze the other combination, and we are going to find out the what are the factors, what are the molecular factors which is going to help to establish the MP diploid, means whenever the two chromosome genomes are coming together, how they are going to stabilize in the further generation from S1 or F1 to the S8 generation or S9 generation, whatever we are having at our hand. So that will be a good study to see the how the chromosomes, how the genomes are going to change from generation wise, from first generation to the 8, 9 generations, and how what are the factors which is going to help to establish that genome together. So that will be the interesting thing to know, because we don't know how what will be the output, but still we are hopeful that we will get some of the genes which is going to be indicate that these are the genes which are responsible for the stability of the genomes when they are coming for the development of polyploid species. So when we develop the integration lines, so there are always the problem of linkage drag. And if you ask the breeders, and they will tell the story how they are facing the problems. Means if they are developing and trying to improve some of the genotypes, but they are getting some of the linkage drag from the wild species or unadapted material. So how we can minimize it? So I was in University of Bonn, and we have done one experiment in 2022-23, where we have treated the population with the gamma irradiation, and then we have analyzed to see the recombination frequency and also to see the breakage in the chromosome, and that we targeted, and we have got that 1750 GY was the most appropriate for the Brassica napus, but we are also thinking to utilize in our breeding population. We have already developed in the at our center, the integration lines, advanced integration line, where some of the linkage drags are available in terms of the late flowering, late maturity with the resistance. So we can break that linkage, and that we can analyze that. So firstly, we are going to do the gamma irradiation, then we are going to generate the population, and in that population we are going to identify the which one is going to perform better and without the linkage drag. So that will be a one strategy to overcome the, one of the strategy to overcome the linkage drag. So that will be also a good strategy to do in our breeding program, pre-breeding program especially. So to summarize my work, what we are doing here, so these are the wild species which we have identified for different traits, for heat, drought, powdery mildew. One of our collaborator is doing white rust, Alternaria brassicula, and aphids they have done. My work is mostly on the heat and drought, and then the pre-breeding research that was published in the different journals. And the same way for the different traits, we have identified some of the integration lines and some of the resynthesized lines, that is the pre-breeding lines for the heat, drought, powdery mildew, and white rust resistance. Already I have shown for the Alternaria resistance we have identified. And also from the mutated resynthesized Brassica, we have identified some of the lines which are performing very, very well. So these are the resources which can the breeder can utilize for the developing the breeding population or the developing the elite lines. So whenever we talk about the pre-breeding, there are a lot of problems, challenges. We have published one paper in TAG, that is the strategies for the utilization of crop oil relatives in plant breeding program. Anyone can go and read that paper. That is, we have articulated the many problems, many solutions. This is from the same paper. So you can see the problem, solution, how we have what kind of challenges we are having, and how we can utilize the advanced techniques to minimize that challenges or that problems. So that will be an interesting reads if you want to learn more about the pre-breeding. So in the conclusion, I can conclude my work like CWRs in progenitor species are important for yield as well as tolerance for different stresses, leads to the sustainable production to fulfill our growing demands of self-sufficiency and self-sufficiency of oilseed. Very necessary in the crops where the target traits are not available in the primary gene pool. If it is available in the primary gene pool, don't think about pre-breeding research in that area. And various pre-breeding methods can be utilized for development of diverse genetic stocks at ICR and IPB Delhi. we have developed and characterized a number of genetic stock which can be very useful for the rapeseed mustard improvement program. Already we are sharing with the different centers, different institute, different universities, different uh, basic science institute for some of the breeding programs, some of the basic science studies, molecular biology studies. Also, we are collaborating with some of the institutions. And also these material can be further utilized to understand the different aspect of intergenomic interactions, nucleocytoplasmic interactions, polyploidy evolution by exploring the combinatorial omics approach. Nowadays, a lot of omics approach are there, so that will be more easy to understand, because earlier what scientists was doing to the pre-breeders were doing, they were developing the variety lines, they were characterizing and they were giving to the breeders for the further utilization. But now we have a lot of tools to understand in each and every step, in each and every generation, how the genomes are moving, how they are interacting, that we can analyze and that we can learn the things, how the breeding program, a pre-breeding program is going on in the one crop, and that can be amplified in the other crops also, and that will be helpful for the understanding of that crop. So we have a lot of population here, a lot of lines, genetic stocks here, characterized genetic stocks, which can be utilized for the breeding program as well as for the molecular biology study. So I thanks my director, my students, and the scientists and staff of ICR and IPB, because they are always helping, and my co-PIs, associate, and my collaborators. A different funding agency, they are funding a lot of, and they are helping a lot that we are doing some of the good research in the pre-breeding. And Dr. Pragati Srivastava for giving me the opportunity to interact with you all and share my work here, and all my students here who are working with me, or you can say that I am working with them, learning together and working to do some basic science in the pre-breeding, because it's very difficult to do the pre-breeding. It's not 1 day job or 1 year job. It's long job to do for the 10 years, 20 years, 25 years. Then you can get something good in from that one. So hope we'll do better in coming future. So thank you everyone. So now if you have some questions, I will be happy to answer, and specific to the pre-bidding or anything, if I can answer, that will be great for me. Thank you
Thank you.
Pragati.
Thank you so much, sir, for such an insightful and knowledgeable presentation. And I'm really thankful and looking forward to collaborate in future for further knowledge distribution.
Thank
Rohit,
you.
Rohit, please open the chat box for questioning, and the participants, please ask question to sir, and you can unmute yourself. You can directly ask your questions to him. Students, please feel free to ask. Don't
Yeah,
hesitate.
if you have any questions about wild species pre-breeding, you can ask, or you want to give some suggestion, that will be also welcome. I will be happy to answer that one. And I have shown one QR code. If you are going to scan that, you can get the information about my group, what we are doing, what we are, and what kind of research we are doing. All you can information you can get from the social media or from the link provided there. So that you can, and even you can write the mail. The email IDs are there. That is the ppeb.nipb@gmail.com. You can write the mail. Definitely, we will come to respond that mail for anything related to my work or our work.
Definitely, sir. I'll share your email to them. So
Yeah,
if they have any sort of queries, they can ask you directly.
yeah. Thank you. So, if there is no specific question,
Sir, there is
uh,
one question from Mr. Vijay
yeah.
Thakur.
How much efficient this pre-breeding can be in systems, sir? Any crop specific precautions we have to keep? Uh, and the pre-breeding is totally different from the other breeding which we are doing in our crops, because it's depend on the crop to crop. Okay. So many things we have to do and we have to learn. Yeah, from the some of the other crops we can learn the techniques how they are moving towards. In system, I haven't worked earlier. I so it might be maybe easy, because these are the deployed species, deployed crops, so you can make the crosses and you can get the seeds. Like in MP deployed crop, it's very difficult to do the pre-breeding. But in case of diploid, it means if you are going to do the pre-breeding in wheat, hexaploid, so you can imagine the problem. But if you are going to do the pre-breeding in the rice, it will be easy way. So this kind of problems you can face if you are going to do the pre-breeding in the different crops. So it's learning by doing process. How we can get the PPT? I will try to make the PDF because it is having some of the data which is not published, and I will share with you all. Okay, I will share with the Pragati and she can share with you all. Okay.
Sure, sir.
How do you manage self-incompatibility when transferring genes from Brassica nigra? Whenever we are trying to cross with the Brassica rapa and Brassica nigra, we are getting the F1 and then we are doing the colchicine treatment. We are getting the that is self-compatible one. Whenever we are going to develop the synthetic amphitriploid, so we haven't got the that self-incompatible. It was self-compatible, like all the polyploid species which we are having, like Zansia, Carinata, and Napus. These are the self-compatible. But yes, even we have crossed the Thoria with Brassica nigra also, because both are the self-incompatible. But we got the, when we are going to do the colchicine treatment and doubling the chromosomes, we are getting the self-compatible Brassica Zansia. So we haven't faced the problem. If there are problem, there is self-incompatibility. So we have to do the SIB matting to maintain the population. Okay, like Brassica nigra we are maintaining, or Thoria we are maintaining. There is no other way to do. BCS, I think, how can regional structure assess these intermediate in material for testing? Yeah, if someone interested to work, they can send the letter to director. They used to forward to me, and there is one MTA. We have to do it. But the intermediate material which we have developed for the synthetic amphitriploid, that we usually not share with anyone, because the ultimate material we can, genetic stocks we can share, but the intermediate material it's very difficult to share because we have planned to work further on that one. So we cannot share that material because that was the intermediate and primary materials. So yeah, the advanced material we can share, and we already shared with many institutions, many scientists for the different work. So for that we are always open. So that is not a problem. But it should be through proper channel that we can do the MTA and we can share. So have you find new source of CGMS in Brassica? I am not working on CGMS or CMS. Some of my colleague was working, and some of my senior were working on CMS system. So I don't know much about this one. So it's difficult for me to answer this question. Okay. So sorry, Rahul. But you can connect Dr. SR Bhatt, was working on CMS, so he can reply. If you can find the email ID, you can mail him. He is a very brilliant scientist, so he can reply on this one. So it was informative session. So just want to ask, what is the best career option for higher ed studies, masters, agriculture, or plant pathology? So MSc in agriculture, you can go for any stream in hardcore agriculture, like pathology is there, breeding is there, agronomy is there. Agronomy is very good option. Horticulture is there, vegetable science is there. And about the location, if you will see the ranking of the any agriculture universities, like institutions, the IRI is the best one for the agriculture studies. You can go to the central agriculture universities. Many central agriculture universities are there. Some of the uh, universities in South, PAU, that's very good option. So it's depend where you want to go and which subject you want to take. So many good universities are. Pantnagar is there, which is having good departments. Some of the departments are excellent. Same way the TNAU, or you can go to Kerala Agriculture University. There that are. But if you want to do the research and you want to do some academics excellence, the IRI is the best place to study. So I think all the agriculture students are knowing that. So sir, if you could design a synthetic brassica from scratch today, which species cross would be the super crop for the next century? In my opinion, I think synthetic hexaploid brassica is having the potential, but a lot of work we have to do, because I have seen some of the synthetic MP diploid or hexaploid from different cross combinations, that is having the potential, but that is having a lot of unwanted characteristics that is not going to yield the giving the yield per se, means seed yield or oil yield. So hexaploid we can try because that is having all the three genomes. So someone can, we are also working on that. We have developed a couple of hexaploids and we are trying to firstly understand, establish that one, then we can think on how we can uh, improve that one. If we can improve that one, so all three genome will be there, all the characteristics of three genomes we can harvest in one genome, one species, and then that will having the potential for the yield as well as for the oil yield and even for the resistance also. So I will suggest, I don't know it will be super crop or not, but we have to test and we have to do the breeding, because a simple hexaploid is not going to work, because I have seen some of the hexaploid species which is not working well. MSc in floriculture is better or not? I have guided some of the floriculture students, not guided, I was in the advisory committee, and they are working very excellent, because there is a lot of scope to do the molecular biology nowadays. A lot of because earlier in the floriculture or vegetable sciences, very basic breeding was done, have been done. So there is a lot of scope to do the advanced tools and techniques you can utilize and do the work in that. And in floriculture you have the scope for the different startups also. So that is good. A lot of students where I am guiding them as a member of the advisory committee, they are coming with some of the problems, and we are trying to give some of the suggestions for one objective or one activity they are doing in my lab or with the help of me. So I have seen that potential in the floriculture also. Floriculture vegetable, because this is the these are the area which will be always evergreen kind of areas, and also the agronomy, horticulture. So you can choose that one if you get good institutions. The best thing is the first and foremost thing is should be the choice of your institutions or universities. That is very, very important. Okay. So
I think sir has covered almost all the questions. And thank you so much, sir.
thank you. It was very good because a lot of participants are there, and hope they have understand some of it. They are from the agriculture or breeding, they have enjoyed it. If they are
Yes, sir.
from other subjects, basic sciences, so they might be liking the photograph which I have shown. So enjoy, have great time, and it was nice to
Yes.
meet you all here virtually. Okay.
Thank you, sir. Thank you, sir.
Oh, fine.
Nandini, I would like you to conclude the
Shall I
session,
conclude, ma'am?
please. Yes, yes, please.
Okay. So thank you so much, sir, for such an insightful and enriching session. Your explanation of complex scientific concepts in such a clear and practical manner has truly made today's session extremely valuable for all of us. We deeply appreciate your time, guidance, and contribution to Krishi Navyug. On the behalf of our entire team and all participants, I extend my heartfelt gratitude to you. Day 17 was not just another session, it was a step closer to understanding the future of agriculture. Thank you so much, sir. Thank you for being with us today.
Thank you. Thank you so much. Thank you everyone for having me here. Bye. See you.
You all
Nice.
can leave now. Thank you for joining with us today.